part 1 video, we have already discussed that immune system can be divided into innate immunity, that is inborn immunity, and adaptive immunity, acquired immunity. The components of innate immunity include macrophages, neutrophils, eosinophils, basophils, dendritic cells. It is a non-specific immunity and it is activated immediately or within few hours of pathogen attack. Main role of innate immunity is to engage pathogen until the weapons, that is antibodies against the pathogen, will be formed with the help of adaptive immunity. It is due to the physical barriers like skin and immune cells like white blood cells such as neutrophils, macrophages, and mast cells. Innate immunity comes into action. Whereas adaptive immunity or acquired immunity, it is specific immunity and is developed when a foreign particle or antigen enters the body and acts on the next exposure. So adaptive immunity is developed when an antigen or foreign body enters the body for the first time. And it is due to lymphocytes, that is T cells, B cells, and antibodies are developed due to adaptive immunity that attacks the antigen. Antigen presenting cells, T cells, which further get differentiated into memory cells, T helper cells, T cytotoxic cells, and B cells, which further get differentiated into plasma cells and immunoglobulins or antibodies. Adaptive immunity is of two types, humoral immunity and cell-mediated immunity. Humoral immunity is mediated by antibodies and it shows a quick response against pathogens. The antibodies produced by the B cells bind to the antigen and they neutralize the microbe. So the antigen receptors present on the B cell, they get activated and plasma cells and memory cells are formed. These plasma cells, short-lived antibodies, memory cells are also formed, which are long-lived, called IgG. These antibodies circulate in the bloodstream, binding with the antigen. Antibodies can bind to viruses or bacteria. They may lead to antibody neutralization. They prevent pathogen from entering and infecting host cells. Antibodies also opsonize pathogen cells, wherein they mark them for destruction by phagocytic cells such as macrophages or neutrophils. Cell-mediated immunity involves cells, mostly T cells. So activation of phagocytes, antigen-specific cytotoxic T lymphocytes, and the release of various cytokines in response to an antigen is the job done by cell-mediated immunity. So once the antigen-presenting cells, they present the cells to T helper cells. T helper cells do a crosstalk with B cells, while they also differentiate and form memory T helper cells and cytotoxic T cells. Cytotoxic T cells are also called killer cells, whereas T helper cells, they are meant for communication with B cells so that antibodies can be formed. This is a network showing how humoral and cell-mediated immunity, they work in combination with each other. The intact antigens, they have a tendency to combine to the B cell receptor present on the B cells, and thus leading to formation of short-lived plasma cells and long-lived memory cells. Whereas antigens which are engulfed and displayed by phagocytes or antigen-presenting cells to the T helper cells, this is a linking step between innate immunity and adaptive immunity, where dendritic cells usually, they act as evidence collectors. They collect evidences in the form of antigen fragments and they present them to the T helper cells. And then these T helper cells can communicate with B cells, resulting in plasma cells and memory cell formation. Also, they themselves get differentiated into T cytotoxic cells and memory T helper cells. These cytotoxic T helper cells further get differentiated into memory T cytotoxic cells and active T cytotoxic cells. The role of T helper cell is to communicate with the B cell and lead to antibody formation. The role of toxic cell is to kill the cells which are infected with the pathogen or pathogen itself. Immune diseases. If the immune system of our body goes against itself and is not able to distinguish between self and non-self, then the antigens which are present on the cells of the body, they are called self antigens. Normal language, antigen should be something which is non-self, but in this case, our body assumes self cells also as antigens. The antigens that enter the body from the environment are non-self, whereas those which are present inside, they're called self. The lymphocytes create antibodies for self antigens known as autoantibodies, and these can cause a lot of autoimmune diseases. The particles present inside the body are not considered foreign particles and are known as self antigens normally, as these are body's particles. whereas non-self antigens are foreign particles that enter the body from the outside environment. But when antibodies are formed in a person's own body against self-antigen, these antibodies are known as autoantibodies. For example, type 1 diabetes or rheumatoid arthritis, in which IgM antibodies are formed against self-IgG antibodies. There are a lot many examples. One more example is Graves' disease, myasthenia gravis. Any disease that results from an aberrant immune response is termed as autoimmune disease. And that's all for the immune system topic of MCAT. Thanks for watching.